Hello everyone, welcome to today's live broadcast, Make Your Clinical Sequencing Count, Technical and Content Decision for Improved Disease Targeted Panels. I'm Rachel Brown of LabRoots, and I'll be your moderator for today's event. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button lower left. I would now like to introduce today's speaker, Dr. Sami Amr, serves as Director of the Translational Genomics Core at Partners Personalized Medicine and as Assistant Director at the CLIA Certified Lab for Molecular Medicine. He is involved in development, validation, and reporting of clinical genetic testing across a variety of disease areas with a focus on hearing loss. I will now turn it over to Dr. Amr for his presentation.
Hello. Thank you for the opportunity to speak to you today regarding our experience with clinical genetics panels. This slide just shows my disclosures. I am an employee of Partners Personalized Medicine's Lab for Molecular Medicine, which is a not-for-profit, fee-for-service CLIA genetic testing laboratory. We do testing on a variety of disease areas, including non-syndromic hearing loss, cardiomyopathy, as well as Noonan syndrome and a whole host of smaller genetic disorders. So in this talk, I just want to really share with you our experiences in running these clinical genetic panels on next-generation sequencing, and what are kind of the things that we've learned across the way, or what are the challenges that we've approached. And really, I've binned it here into two different major concepts that I'm going to be talking about or covering in a little bit. Some are the technical considerations to think about, which are things in trying to identify variants, making sure that they're true variants and issues related to that. And then the other piece that's also related to these clinical panels is the interpretive and analytical challenges that we've encountered. And we'll talk about that in a little bit. So these are just the different sequencing testing platforms that we've used over the years in our laboratory, and we continue to use. Of course, everybody's familiar with Sanger sequencing. It's known as the gold standard. Then we moved to larger panels using resequencing arrays, and now we're all using next-generation sequencing panels. And here we're comparing these three testing platforms on several parameters, including target size, cost, and turnaround time. Of course, Sanger sequencing is optimal for smaller gene panels or one gene test. You can do it fairly efficiently, fairly quickly. Resequencing arrays allowed you to expand the number of genes that you can do at once. And of course, next-generation sequencing, the major advantage is that you can do many number of genes across a large number of samples all at once. And this is shown here by the target size. The cost of Sanger sequencing is lowest when you're looking at a single gene. If you're just sequencing a single gene for one patient. However, for when you expand to a large number of panels, a large number of genes, or a large number of patients, then next-generation sequencing is more advantageous and can be cost-effective if used in that manner. The turnaround time for a single reaction, when you're looking at Sanger sequencing, it is much lower. It's 1 to 2 weeks. Resequencing arrays is somewhere in the middle. That's 6 weeks, and next-generation sequencing, when these slides were made, it was 7 to 8 weeks. But as we all know, with the recent advances in both the reagents and the bioinformatics, this can be up to 1 week nowadays. So it's definitely improved over time. And now the advantage of next-generation sequencing that we've encountered is that it can detect all types of genetic variation. And here we show the different types of the variations, just screenshots from different applications that we've used it for. So here we use, this is just a single nucleotide variant that we've used. Let me see if I can, sorry about that. This is a single variant that we've used. This lower one here shows a indel that we've identified an intronic region of a gene, and it also can do copy number variation as shown in this plot here. So here we detected a heterozygous H2A 2 exon deletion, or a homozygous H2A 2 exon deletion in an individual with non-syndromic hearing loss. The sensitivity for SNVs is very high. It's comparable to that of Sanger sequencing, as well as the specificity is very high. It's a little bit lower for indels. It's a little bit harder to detect indels using next-generation sequencing, and that is it depends on the size of the indel and the location of the indel. So if it's in a repeat region, or if it's very large, then it might be more difficult to detect these indels. CNVs can be detected, and the way that different labs do it is that they're looking at CNVs is detected using the coverage of a region compared with the coverage of the region from a different sample. So you're taking each one of these reads essentially, or the coverage at a certain region, and comparing it and seeing whether whether the ratio would indicate that there's a copy number loss or a gain based on coverage metrics. I don't want to get into that too much, but I do want to tell you kind of the issues that have come that we faced as a result of just using that approach to be able to identify CNVs. So the sensitivity for copy number variant detection is high using our experience. However, what we found is that also the specificity is low. So we're seeing a lot of false positives, and I'll show you a few on the next page. So this is the output that we get from copy number variants using our algorithm or our software tool using next-generation sequencing data. So the panel on the left shows a heterozygous otoferlin deletion of 47 exons of this gene, and the right panel shows a homozygous 2 exon deletion in the H2A gene, which I showed earlier. However, you're also seeing these small, I'm going to try to point them out, these small calls that are shown here. Are these truly gains or losses of other loci within this gene? What we follow-up testing confirmed that there is a high false positives, especially with single exon calls, and you can see that there's a lot of kind of noise associated with this. And when we followed up on all these single exon calls, we identified them as false positives. So it does still have a low specificity, and you do need a secondary assay to confirm CNV calls. The other piece is that these two plots look really nice. However, the success of the algorithm to work and to be able to make these calls is dependent on batch effects. So every single batch may behave differently, and there's a lot of bench process. So the hybridization of the NGS capture has to be done properly and has to be very efficient for it to work. So we find often that hybridization may be sufficient to call SNVs and to call indels. However, the CNV calls is too noisy to make a call, and we end up failing the CNV caller while passing the SNV and the sequence detection. So we use next-generation sequencing panels for all our disease areas. This just shows you the list of genes that we use it for our non-syndromic hearing loss and hearing loss related disorders gene panel. This is a 70 gene panel, 70 gene targeted panel. that includes genes associated with non-syndromic hearing loss, but it also includes genes that are associated with syndromic hearing loss in syndromes that appear initially as non-syndromic hearing loss. So the initial presentation or the first presentation that the physician may see is non-syndromic hearing loss, and then other features may develop over life. And so I just wanted to share with you results from a small cohort from this panel that shows sort of our experience with this. From a cohort of 87 positive individuals with hearing loss that were run on the autogenome panel, there's two things to really note from this slide. One is that the genes that are causing hearing loss in this cohort are many. So there's definitely genetic heterogeneity in this cohort or genetic heterogeneity that causes hearing loss. And so it's not enough to just go after a single gene. It's really important that we broaden our tests so that they include a large number of genes so as to be able to identify the causative variant. The second piece to really think about is shown in this table down here, which illustrates that the causative variants within these genes are also broad. So you can have missense variants, you can have nonsense variants, you can have small deletions or insertions that cause hearing loss, but you can also have a large number of causative variants or copy number variants. About 20% from our cohort, 20% of individuals had a copy number variant that was causative to their hearing loss, which shows not only how important it is to be able to carry out testing, but also to be able to detect copy number variants in these individuals, because we may be missing a large number of a large number of causative variants if we don't include copy number variant detection within our next generation sequencing platform. So to be able to do CNV detection, we know that there's a high false positive rate. We know that that it's also doesn't work for every batch. So how can we supplement, or how can we make sure that we do cover CNVs in whatever testing strategy that we use? So there's often what different labs have done, including ourselves, is that we've used secondary assays for confirmation of CNVs. So if we detect a CNV on the next generation sequencing platform, we might follow up with an NLPA or digital droplet PCR to confirm whether that variant is true or not. Another option would be to supplement with a different CNV detection platform. If we find that the false positive rate or that the fail rate for the CNV portion of the NGS assay is too high, then we might opt on not doing CNVs on the NGS platform, but carrying out a supplementary assay next to the NGS data to make sure that we cover to make sure that we're covering copy number variants. I know a lot of labs are doing that. They're offering exome sequencing on NGS, which covers sequence single nucleotide variants as well as indels, but they're also carrying out exome level array CGH, for example, to be able to detect CNVs on a separate platform. So here I want to just give an example of one of the regions that's very critical to being able to identify copy number variants in hearing loss, and how we've kind of come up with a secondary assay to approach or to address the copy number variant issue. Essentially, there's a region in upstream of GJB2, a very common gene that's associated with non-syndromic hearing loss, which is subject to large number of deletions. So there's two common deletions that are shown in this figure, and both of these deletions lie upstream of GJB2, located here. These deletions essentially cause repression of expression of GJB2, thereby causing hearing loss. Our normal GJB2 strategy is to be able to sequence GJB2, and we have a couple of gel assays that can detect one of these two deletions, the more common one. So we can't detect both of these in the method that we're doing it now. Alternatively, we also have probes that cover this entire region from GJB2 all the way to this upstream gene CRY. I can see it on my, but upstream of GJB2, and that cover the entire region next to it, using probes designed to be able to identify using the NGS data. However, you have to run the NGS data, the NGS platform, to be able to detect the variants. So this is kind of the design that we've used. So the VisCap target, or the NGS targets, each one of these right here represents probes that we've designed to be able to hybrid capture these regions that lie upstream of the GJB2 gene. So we're able to detect these deletions. And while those two up here in the first graph, or in the first figure, represent the most common deletions, there are other deletions that have been identified that are also important to be able to detect. And so what we've designed are a number of ddPCR probes that essentially 5 ddPCR probes that sit in one of the several, well, they sit outside as well as inside the deletion regions. And using ddPCR probes, we're able to run the CNV assay without having to rely on NGS data, which may fail or may cause lots of false positives. In addition, GJB2 represents, or variants affecting GJB2 represent 25% of all variants that cause non-syndromic hearing loss. So some labs may opt to do GJB2 sequencing first using Sanger sequencing because it is cheaper for one gene, and then reflex to next generation panels if the GJB2 Sanger sequencing test was negative. And so you wouldn't be able to pick up all these deletions unless you're running the NGS panels. So what we did is now we're running the GJB2 test, and we're using the ddPCR assay as a way to be able to detect the CNVs. And that allows us to do two things. We mitigate the failure rate of the NGS CNV assay. ddPCR has a lower failure rate than the NGS CNV assay, and so that way we're able to capture these copy number variants fairly efficiently. As well as the second piece is that the NGS assay is much more expensive than the cost to just run the ddPCR. And here we just show what the price is from our lab of the two different tests. So to be able to run the NGS platform, whether you're just doing it for copy number variation or if you're doing it to capture sequence variation as well as the NGS variation, it costs anywhere from $2,500 to almost $4,000, while the cost to run the ddPCR assay is under $1,000. So there's other limitations of NGS panels that are also not associated to CNVs. This includes GC-rich regions. These are regions that are difficult to capture using the current reagent technology. And there's issues with, as I mentioned earlier, being able to identify larger indels, being able to look at repeat expansions, and the issue of pseudogene contamination due to high homology. So there's a gene of interest that has a pseudogene that shares high homology with that gene of interest. It's difficult to kind of identify whether variants that are seen in that gene come from that gene or if they come really from the pseudogene. And we'll talk a little bit more about that a little bit. The issues of large indels as well as repeat expansions, we're slowly overcoming that with longer read technology as well as better bioinformatics analysis to be able to pick up those things. So talking about, so I want to focus on pseudogenes and the issue related to that. And a great example of that is the STRC gene, which is one of the genes that also has contributes to non-syndromic hearing loss. About 10 to 50% of all individuals with non-syndromic hearing loss have a deletion in this STRC gene. But the issue with being able to test for the STRC gene lies in the fact that this gene, which is located here, has a pseudogene, which is located upstream of this gene, that shares about 99.8% homology with the actual functional gene, and this includes exons as well as introns of that gene. And like I mentioned before, however, we need to be able to capture this to really increase the clinical sensitivity of the assay, since many individuals who have hearing loss have variants or copy number variants in this gene. So this diagram here up here illustrates the gene. This part of the gene, which is shown in black, is actually at 100% homology. The 3' end of the gene has lower homology, but it is still very high. So to be able to let's see if I can. So to be able to capture this gene, if we run normal Sanger sequencing of each individual exon, or if we do regular capture and NGS, what happens is you get outputs like these two right here, which is pseudogene contamination at any particular position. So it's not clear if the variant comes from the pseudogene or if it comes from the actual functional gene. And to mitigate that, one solution that we found is to run long-range PCR of the entire region using primers that are unique specifically to that gene, and that basically border the gene from the 5' end and the 3' end. And then subsequently you can either run it using Sanger sequencing, or here we show that we're running it using NGS. So you can literally take that amplified long-range PCR fragment and throw it onto the MiSeq or the HiSeq or into whatever capture you've done initially, or you can also just run it as a separate Sanger assay. And this way we've been able to identify true variants in the SDR gene and eliminate the pseudogene contamination of those genes. And we published our results in Journal of Molecular Diagnostics shown here. The other issue associated with SDRC, you also see issues with being able to identify copy number variants. And this is, again, important for SDRC because there's a large number of variants in SDRC, specifically copy number variants seen in hearing loss cohorts. In the top panels up here, you can see what a non-pseudogene gene CNV output would look like. So a heterozygous deletion would be near the -1 on this log2 scale. A homozygous deletion would be down here at the -2 or -2 on the log2 scales, and this is typical of what you're seeing. However, because of the pseudogene contamination, it also affects the detection of CNVs and being able to call the zygosity of that CNV. So essentially, this is meant to kind of illustrate what's going on with the pseudogene. Instead of having 2 copies of each gene, you have 2 copies of the gene, but you also have 2 copies of the pseudogene. And when you lose, in a homozygous deletion, you lose 2 copies of the gene, but you still have 2 copies of the pseudogene. Therefore, it looks like a heterozygous deletion for regions that are high in homology. And so when you run the SDRC on VisCap, you see this kind of waterfall, this waterfall shape, or this waterfall looking graph. Regions that are up here, where shown in the arrow, are regions of high homology, and regions down here are regions of that gene that have low homology and are more representative of the homozygous deletion that you're seeing in that gene. So again, to be able to, while this kind of, it flags the CNV that yes, it does exist in your sample, a follow-up test using unique primers via ddPCR is needed to be able to verify whether this is a heterozygous deletion or homozygous deletion and what regions it does affect in the SDRC gene. So just to kind of summarize what we've been talking about in terms of technical challenges, it's important to have secondary assays to confirm variants We use currently Sanger sequencing for single nucleotide variants, and we've done MLP or digital droplet PCR for CNVs. And it's important to consider supplementary assays for variant detection, and this is true especially for copy number variants, as we know that NGS technology is not exactly where it needs to be for clinical copy number variant detection. And so for high homology regions, for single nucleotide variants, as well as copy number variants, we use long-range PCR for the single nucleotide variants, and we're using ddPCR or other methods for CNV detection. Okay, I want to shift a little bit in terms of what we're talking about now. So we've looked at technical considerations in running these clinical genetic panels. I also want to talk about the interpretive and analytical challenges that that we've encountered while expanding onto the NGS platform, adding more genes and running them across large cohorts. And there's really two major contributors that we faced, or two major challenges that we faced, and the challenges come from two things: the large number of novel and rare variants that we've identified in these cohorts, and the growing number of genes on NGS panels. So in the slide a few slides ago, I showed you 70 gene panel for hearing loss. Well, just last week or 2 weeks ago, we launched a new non-syndromic hearing loss panel that has 87 genes. And so the number of genes that physicians are asking for, that researchers come up with in terms of their association with disease, is ever expanding. And more so, we're even moving from the paradigm of looking at one disease, and now we're moving towards the exome. So we're looking at all 30,000 genes, or the medical exome, which is 5,000 genes. But it's just an expanding number of genes that molecular geneticists are having to look at to try to identify and assess variants that are related with a certain gene, with a certain disease. And certainly this is one of the biggest bottlenecks in being able to provide a test with fast turnaround time. So this figure illustrates the number of variants that our lab encountered over 15,000 probands that were run here, and this is for all disease areas that we've looked at, including hearing loss, cardiomyopathy, myopathies, Marfan syndrome, etc. And the important thing to see here is that is shown by this first by the first column here, which is the majority of the variants that we've seen, whether they were pathogenic, whether they were variants of unknown significance. They were all seen in one individual or one family. So these are variants that haven't been seen before, haven't been reported anywhere, and we're having to go in there and try to, based on the information that we have, which is very little, to try to classify these variants and to try to understand whether they are related to disease or not. There are some variants that are very common that are shown here that we know about and are easy to classify, but for the majority of variants, they're unique and they are rare, and obviously you can imagine that every variant requires scrutiny, and a lot of time is spent to try to interpret those variants. Now, the other piece that complicates this is that the number of genes that are being discovered and that are associated with disease is increasing. So this graph is taken from information from HGMD, the Human Gene Mutation Database, and you can see that every year a larger number of genes are deposited into this database and are associated with disease. Now the question is, how are these genes associated with certain diseases? And what is the evidence that is used to kind of promote and to kind of support those gene-disease associations? And here's a list of, on the left is a list of different databases and sources that we look at when trying to determine whether a gene-disease association does exist. Now, the issue with this is that a lot of these databases, they deposit information, they don't check the veracity, or they don't check the level of evidence of these gene-disease associations. So it's not always, it may be one paper, one publication looking at one family somewhere that creates or that indicates or suggests that a gene-disease association. However, is it truly there? That remains to be seen, and it's the job of the genetic laboratories to look into that to determine whether these gene disease associations truly exist or not. So the best way to kind of demonstrate the issue of gene disease associations is with a case that I'm going to show here. So part of our original 70 gene panel, we included 2 genes that are shown down here, GJB6 and MYO1A, and I'll be talking about these in detail. However, so we'll start with this case. It's a 7-month-old male who presented with congenital profound sensorineural hearing loss. And when we ran this the sample for this patient on the autogenome, we identified 2 variants. We identified a frame shift variant in the GJB6 gene, and we also identified a missense variant in the MYO1A gene. So the question was, do we think that either of these variants are related to this individual, to this patient's hearing loss, or is one of them causative for the hearing loss or not? And so that's kind of the novel variant assessment process that we undergo every time we do clinical testing. So we'll start with the GJB6 variant. So GJB6 variant associates to a very well-known hearing loss gene, GJB2. It's supposedly co-localizes to the cochlea, to the inner ear, and might create plays a role in gap junction formation. We don't have sufficient evidence to really understand what it does and what its true role is in the inner ear. Now, it has been associated with several different diseases based on the literature and based on OMEM and different databases. And it's one of its disease associations is with hydrotic ectodermal dysplasia. 2 missense variants have been identified individuals with that disease. And it seems that there's several reports that support that. So we're pretty, pleased with the evidence that associated with hydrotic ectodermal dysplasia. However, keep in mind that our patient doesn't have hydrotic ectodermal dysplasia. They presented with non-syndromic hearing loss. It's also, GJB6 has also been associated with autosomal dominant hearing loss. There's 2 different reports that make that suggestion. In one report, it's a missense variant that has been associated with autosomal dominant hearing loss. In the other, it's a frame shift variant, shown here. In these 2, however, the evidence is just based off of one family. It's one report for each one of these. And if you look closer at these variants and use additional data, we found that these variants both are present in the population. So they're found in exact. So it's, we don't know if this gene is related to autosomal dominant hearing loss, but we don't think that the information we have based on these 2 variants is sufficient to make that assumption. Now, what has been established is that GJB6 deletions, large deletions, has been associated with non-syndromic hearing loss. And we've seen this figure before, earlier. We know that deletions that affect GJB6, we know that deletions that are upstream of the GJB2 gene affect the transcription of GJB2. So it's not really deletions of GJB6 that are causing hearing loss, it's rather than deletions that lie upstream of GJB2 and affect its expression, which cause hearing loss. So again, the gene disease association with GJB6 is not really understood, and so it becomes difficult for us to try to identify or try to classify the variant. So in this case, it's a frame shift variant. The evidence suggests that it hasn't been reported in individuals with hearing loss. This specific variant has been seen in the control population, although it's a very low frequency, so it's possible that it's still causative of something. And because it's a frame shift, we know that it may cause a loss of function of the protein. And so it will have a detrimental effect on the protein, so it, we can't rule it out. However, because we don't know if JGB, like the variants that affect GJB6 specifically cause non-syndromic hearing loss, we can't say that this variant is pathogenic for the hearing loss seen in our patient. And so this variant we classified as a variant of unknown clinical significance. What about the second variant that was seen in this individual? The second variant was a MYO1A variant, missense variant in exon 3. Here's what we know about MYO1A in both Omen and in Gene Reviews, two very well-known databases that curate this type of information. Both associate MYO1A with autosomal dominant deafness. And you can see that here, Omen as well as Gene Reviews. So we looked at that specifically what drove both of these databases to include that gene disease association, and all of it is based on one publication, which found MYO1A variants in a large cohort of hearing loss individuals. So we looked at all the variants that were listed in that publication. The publication was published in 2003, and since then we've had opportunities to sequence a large number of individuals, a large number of control populations, and just get more variant information on common variants. And what we identified is that a large number of the variants that were identified in this patient and in this cohort were either found in control populations, making it less likely that these variants are causative for hearing loss. In one case, one of the variants was found at 1%. The majority of variants that I classify when I'm looking at hearing loss, variants identified in hearing loss patients, is that anything that's above 1%, albeit except for a few common hearing loss variants, they're likely to be benign. So anything above 1% is likely to be benign for hearing loss. Another variant was seen at 3.8% of the population, even more so less likely to be causative for hearing loss. So a lot of these variants were seen in control populations, so so that makes this gene, the gene disease association between MYO1A and hearing loss less likely, because this is the only piece of evidence that we have, and now we can exclude it because these variants have been seen at such high frequencies. This is not to say that MYO1A is not causative for hearing loss. It's just, given the current evidence that we have, we need more information to know whether truly this gene disease association exists or not. So how did we classify the missense variant that we identified in our patient? It was not reported in any other individuals that we've, or in large populations. The computational prediction programs, they look at missense variants and we look at conservation and a lot of other elements to try to understand whether whether a missense change is actually pathogenic or not. They didn't provide any information or did not provide evidence for or against pathogenicity. Okay, sorry. So we classified this variant as a variant of unknown clinical significance. So to summarize this case, we identified two variants. Both were classified as variants of unknown significance, and so the report went out as an inconclusive report. Now this, in my opinion, is the reason it went out as an inconclusive report is because we identified two variants in two genes that we did not really know whether the gene disease association existed for these genes with hearing loss. And it becomes not an issue of the way that we classify the variants, but it becomes an issue of whether to include these genes on hearing loss panels. We don't know enough about these genes to associate it with hearing loss. They only add confusion to the report. They definitely take time to interpret and analyze. Sanger follow-up has to confirm all these variants that we identify. So it just takes more time, more energy, more greater cost to be able to go after these variants, when in essence, we don't even know if they're truly associated with a disease. So what we did was, we found that there's a large number of hearing loss genes that have been associated based on evidence that was not strong, that was not compelling. And so what we went back and did, we looked at all the genes that are on our hearing loss panels, as well as other commercial laboratories who offer hearing loss testing, and we binned the genes based on evidence into one of these 5 categories, whether there was an association or if there was not a strong or compelling association. And the way we did that was we looked at the variants that were identified in these reports, that were identified in these genes, and whether those variants were segregated with a disease, whether there was any functional analysis or in vitro or in vivo mouse models or animal studies that were done to knock out the gene and see the effect of that gene in animals. So we kind of used all the evidence that was available to us to kind of either classify the gene as having a strong association or a weaker association. And when we did that, we looked at 145 different genes that are available in our panel as well as other panels, and we found that 54 of those genes, that's about 37% of all the genes that we looked at, did not have strong evidence or compelling evidence to be called non-syndromic hearing loss genes. So we're not saying that these 54 genes are not related to hearing loss, we're just saying that the evidence would lead to inconclusive reports, as I've shown earlier, and it just leads to more complication in testing and interpretation. So the idea would be to be able to exclude these genes from future clinical gene panels so that you're able to provide a better clinical sensitivity and clearer reports to the patient and to the physician. And one way to do that is for clinical molecular laboratories to share information on variants as well as genes and to share their experiences in trying to interpret these genes and in trying to interpret these variants. One such effort there's several efforts out there. One such effort is carried out by the Clinical Genome Resource Task Force. It's headed by our laboratory director here, Heidi Ream. And essentially the idea is that all laboratories share the variants and their classifications and what genes they've classified, as well as the way they're looking at gene disease associations and what they've included on their gene panels. And this will help us grow our college knowledge database so that we're able to better include certain genes on different hearing panels, whether it be hearing loss or cardiomyopathy or any disease area, but also to systematically classify variants the same way and use data across labs to be able to support that classification. So to summarize really quickly, when our experience with clinical genetic testing panels led to some challenges both on the technical end as well as on the interpretive end, and it's important to really consider both of these when approaching next generation sequencing panels, when trying to develop new panels, when trying to add new genes, that everything is looked at, and everything is considered, in terms of copy number, variant detection, as well as pseudogene homology for the technical side, but also, really looking at genes and gene disease associations critically to assess whether they should be included on panels or not. So with that, I'd like to stop here and just give an opportunity for us to have a discussion if you have any questions related to this or related to NGS clinical gene panels in general. I also want to very quickly thank the organizers of the event, as well as Rachel and Don for hosting me and allowing me and giving me this opportunity to speak to everyone here today.
Thank you for that informative presentation. Before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. And our first question is, what about using nanostring to confirm CNV results?
Yes, I've heard of several laboratories using nanostring. I think I don't have any experience personally with it, but I think any type of secondary assay that has been validated could be used for copy number variant detection.
Okay, so this next question, it was posed kind of differently. It reads, used to Sanger sequencing, will my lab have to go NGS sequencing?
Well, it all depends on what kind of testing that you offer. So, to go to an earlier slide, if you are interested in, it really depends on what kind of testing that you do. So if you're only doing single gene testing, if you're doing a handful of genes, then Sanger sequencing still makes sense. It still has a fast turnaround time and it's relatively cheaper to other methodologies. However, if you're expanding to 70 gene panels, as we are doing with hearing loss, if you're expanding to the exome, which many labs are doing now, to be able to capture that, it's virtually very cost ineffective and very time consuming to do that with Sanger sequencing. So I think it depends on what the strategy of the testing in your lab is and the number of genes and the diseases, of course. Again, if you're looking at a single disease that has a single variant or a single gene that's associated with a disease, for example, cystic fibrosis, you can do fine by just sequencing the CF gene. However, if you want to do a pulmonary panel, which there's more than 50 genes, including the CF gene, then no, next-generation sequencing would be the way to go.
All right, so it looks like those were all of the submitted questions. And we'll go ahead and wrap this up. Today's webcast will be available for on-demand viewing through November 2015. You will receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time. Goodbye.